My name is Alex Pulakov. I'm from this beautiful place. It's a whaling village on the north shore of Long Island. We're trying to crack the brain. So, not literally. So, I realize that not everybody is an expert in the brain, so I'm gonna just briefly describe how the brain works. Less than 5 minutes. So this is basically how it works. So a brain is composed of neurons. Neurons have cells, which are giant capacitors. They look like octopi. They have two types of branches. So there's something called dendrites, which are passive waveguides. They receive information from other neurons. That information travels to the cell body, soma, where it's composed, it's added up, and then at some point, neuron makes a decision to emit something that is called a spike. The spike is like a soliton, which is basically the membrane will be short-circuited very briefly for about a millisecond, which can propagate at enormous speed along something called an axon. A neuron usually makes only one axon, so there's only one decision that it makes. And then it arrives when the spike arrives at something called a synapse, which is a connection between two neurons. There is some kind of molecule will be spilled from the synapse, and it will diffuse to another neuron, and it will produce either excitation or inhibition in another neuron. And that's how information spreads through our networks, and that's basically how the brain works. So you've already heard everything. So another interesting factor is, I borrow it from the title of the book of Sebastian Sung, is that the current hypothesis that what you are, your personality, your memories, your skills, is stored in the connection matrix between the neurons, and which is illustrated, which is schematically shown here by those diffusion tract axons. Those are like connections in the white matter. So the number of connections, of course, in human cortex is enormous. We have about 10 billion neurons in cortex. Each of them makes about 10,000 synapses. There is a lot of information we can store. So I would like to make this very simple back of the envelope estimation. What is exactly the amount of information that we can store? And we have like 10 billion neurons. We have 10 to the 4th synapses per neuron. I learned this formula that the amount of information is just the entropy is just a log of number of combinations. I can find the total number of matrices with those parameters by binomial formula. And if I do this calculation, I will get something like 400 terabytes. And I understand that it's not very easy to many people to handle terabytes, so I converted it to movies for you. So it amounts to about 45 years of continuous high definition video. So our cortices can store enormous amounts of information, like your essentially entire experience in 45 years. And so it's, I mean, this is the back of the envelope estimation, but the thing is that there's a logarithm there, which, as we know, is 1/3 power. And, so it's very shallow function. So even if you make a mistake by a factor of 2, it doesn't make much difference. Another thing to put this in perspective is the amount of information which is present in the genome. Genome contains about 1 gigabyte of information, which is about 5 orders of magnitude less than this number. Just again, in terms of perspective, it's about one hour movie, right? Like high definition video. So it's like 1 hour versus 45 years. But the interesting thing is that the brain cortex is created from the genome, from the information, from the blueprint which is contained in the genome in the course of neural development. How is this possible? It can be viewed as some form of paradox, and this paradox was discovered many times throughout the years, beginning from 1963 by the guy who actually discovered the modern rules for neural development. He was a biologist who was actually informed about information theory, and it was also discovered by us at some point. So it's an interesting observation. Like the good news here is that whatever differentiates us from mice or from monkeys, for example, is contained in a short instructional video, which is about 1 hour long, right? So in principle, it's easy to explain, right? It should be easy to understand. So the bad news is that there is some kind of discrepancy here. So how can we create this functioning brain from such a small blueprint? The paradox is, of course, is a little bit contrived because those are different quantities. So what genome contains, it contains something called Kolmogorov complexity. It contains a program, right? So although the information and Kolmogorov complexity are both measured in bits, so those are completely different quantities. You can have very short program that creates a very complex structure, such as, for example, you could say that this is a network which is set up randomly, right? It requires very little information. But we know that brains are not created randomly. So the idea is that genome contains some kind of instructions which were inferred in the course of evolution, which are really important. And that is what is called genomic bottleneck. And just to illustrate why it's called a bottleneck, there is this picture. So remember, there is this red arrow where the genome is expanded and it creates our brain of a newborn child. But there is also another arrow, which is here blue, which is when the brain is actually imprinted in the genome. It's shrunk back in the genome. And what I mean here is not the evolution, I mean evolution, right? So this arrow is produced by evolution, whereby the most fit brain will be represented in the genome. I don't mean that there is like the information from the brain somehow goes into germline, or there is some kind of Lamarckian evolution. It's just that this blue arrow represents a natural selection. Okay, so the idea here is that in the course of evolution, brain has undergone millions of cycles of expansion and contraction, So each generation represents one of those cycles, and so which is an interesting thing, and the idea is that that's why we call it a bottleneck, right? So each generation, brain structure is put through a bottleneck, through a narrow information channel, and so one possibility that this bottleneck is just a bug, right? So we have to deal with it somehow. But an interesting hypothesis to pursue, which I'm going to argue for in my talk, that this is actually a feature, because imagine that you need to pack like a bag, but you have only a small bag, but you have to put your entire life's experience into that bag, and your enormous cortex, all of your memories. So you will probably be very careful about what you are going to put there. So this is a constraint that we argue is important, and therefore the idea is that evolution has shaped the brains in such a way, and it has shaped the genome in such a way that we keep only important features that will generalize, which will help our offspring to survive. Questions?
Yes, probably naive one. Why brain and not, let's say, giraffe's neck or other selection principle that passes on this information?
Well, giraffe's neck is a simple structure. There's not much information there. It's probably like a few bits of information is easy to set up. But yeah, so the idea is that there are a lot of giraffe necks which are stored there, which we want to discover. So there are a lot of simplifying principles that have to be operating in the brain. So it's not just a amorphous network that learns, like in AI, for example, that learns everything. It's shaped in the course of evolution to learn only specific things. Yes?
Is this similar to the idea of a grandmother neuron?
The brain is what?
Is this similar to the idea of a grandmother neuron? One maps to this mesh.
It's not a grandmother neuron because there's still a lot of information there. It's just, yeah, I mean, it's similar, but it's not like there's one neuron. It's like 1 gigabyte of neurons. Like in terms of amount of information that you have in the bottleneck and it's narrowing, there's a lot of information, just not one neuron. But probably a grandmother neuron would be one of those solutions, right? So it's one potential solution. We just want to be able to discover them. So this is the genomic bottleneck hypothesis, is that a genomic bottleneck had led to the distillation. So this process of cycling brain through the bottleneck, the distillation of important information about statistics of the outside world, and to emergence of general intelligence. So it's a hypothesis that we're trying to test. Of course, it's difficult to test evolutionary hypothesis. And so I'm going to try to test it using tools of artificial intelligence that I'm going to describe you in the next half hour or so. So one of the ways how question.
So there's also epigenetics like methylation. Is that also taken into account in this 1 gigabyte?
It's a great idea. So of course, there is also epigenetic information which is stored in the cells, which is passed along the offspring, but it's probably less than 1 gigabyte. It enhances it probably by a few percent, but it doesn't really resolve the 5 order of magnitude difference. That's what we think. So, of course, like one of the things that how this bottleneck can work is through, for example, creation of the learning rules that will operate in the course of our life. So those learning rules could be simple, but another way is like in many organisms, some behaviors which are some organisms are created, they're already perfectly capable of doing very complex behaviors right after birth, right? And those behaviors must be stored in the genome. And I'll just argue that those behaviors can be very complex. So I'm just listing some of them. Of course, many of us can relate to this baby geniuses, right? And the newborn turtles, which is my favorite example. So the turtles are born in the sand, close to the beach. They wait for the darkness, for the nightfall, because otherwise, if they pop, so you see this turtle is waiting for the sunset. Otherwise, they'll be eaten by the predators, and then they come out from the sand, and then they run to the sound of the seashore. Of course, they have never learned this behavior, right? It's totally imprinted in the genome. So many animals are capable of walking. That's like this newborn deer right after birth. They follow their mother. So like the spiders can make their webs, birds can fly. And another interesting phenomenon is the human capacity for language, which according to Chomsky is some features of it are imprinted in the genome. So that's why it's easy. We can learn languages, so very few examples. So those are some examples. So we decided basically to pursue this hypothesis just using artificial intelligence to see if we can create like an artificial intelligence analog of this genomic bottleneck. But before we do that, I just want to review the general idea of how is it possible, how do the genes, how do they create connections? So what is the mapping between this one-dimensional structure of DNA and the complex topological structure of connectivity in human cortex? And the idea how it was created was hypothesized by this person whom I already mentioned. He proposed the information theoretical hypothesis about bottleneck, the first, Roger Sperry in early 60s. So the idea is this. It's very simple. So I'll just read it. The cells and fibers of the brain must carry some kind of individual identification tags, presumably cytochemical in nature, by which they are distinguished one from another, almost in many regions to the level of a single neuron. So the idea is that neurons will emit some kind of molecules. They will carry and emit a molecule, and the other neuron is capable of smelling that molecule. I'm just translating this sentence to like modern English. and recognize, grow in the direction of the molecule that they want, and they will avoid molecules they don't want. And by using this kind of cellular sense of smell, they're capable of finding neurons that they want to connect to. So that's the hypothesis. And 30 years have passed since that hypothesis was formulated, and some of the molecules were discovered. And I'm going to give you an example of this system where it's basically the connector from the eye to the brain. So like an eye, we have about 1 megapixel in the eye. So there's about 1 million fibers that grow out of the eye, and they have to connect somewhere to the brain. So how do they do that? That has been figured out. So this is an example. So this is a cross-section through the mouse eye. This is like a pupil, you can see. This is retina, retinal ganglion cells. Those are the connections that grow out. And so this is the surface of the retina. And what is shown here is this density of the protein, which is called FA5, which if you look carefully, it has a gradient. So it's high here, close to the temple, and it's low here, close to the nose. So basically, the horizontal coordinate of the eye is set up by the density of this protein, which is roughly exponential, actually. So the model works like this. So this is in the eye, there is this protein called FA5, and in the target of the eye, which is called superior colliculus. I'm sorry, there is some Latin naming here. There is a ligand for this protein, and the ligand has also this graded expression, and the neurons, they grow out of axons, they grow out of the eye, and they are capable of distributing themselves along this axis of the target of superior colliculus. And I recognize that this is all very complicated, so I just formulated it in terms of electrostatics model. So imagine that this level of this protein is just the charge UA, and the level of this ligand is the electric potential phi A, and you can just like write a Hamiltonian for this interacting system. So this would be a sum over axons of the charge associated with each axon and the potential where this axon terminates, and of course, like all axons, because like this interaction is obviously repulsive, all axons want to be here. They want to slide to the point of minimum potential energy, but that is forbidden because they have there are actually interactions. There's exclusive repulsion between them, so they cannot occupy the same space, right? So it's actually an interacting system. So I'm not showing here the interaction part of the potential just for simplicity, but the reason why they cannot slide all here is because they repel each other.
Some chemistry that does it for it.
It's a chemistry, yeah. It's a very funny chemistry, but I mean I can talk about it for like an hour, but I'm not. So just assume that there is like Misha was describing, there is a repulsion between them. So it's an interacting system. And like if you just minimize this Hamiltonian, you will realize that they will distribute themselves along this axis. So the highest charge axons will end up here, and the lowest charge axons end up here. And imagine that you just like minimize this like using some kind of Monte Carlo simulation. So of course, the question that you can ask is what happens to the vertical axis? So how is it? It's only 1D, but how do we do the second dimension? And of course, for the second dimension, there is a second molecule, which is called FB, which is just another charge of a different color, which is distributed like this. And for this charge, there is also another potential called FNB, and that's from this layout, you can see there is actually attraction between the FB and FNB, and you can just add another part to the Hamiltonian with a minus sign because repulsion. So this is basically how this connector, like a million wires, grow out of the eye. There are actually 6 targets, so this happens 6 times in the brain, but in a mammalian brain. But the overall idea, I mean, you understand the overall idea is that there is a system of charges and there is a system of potentials, and they're organized, choreographed in a complicated manner that can create a very complex connectivity. Yes?
But then this would give you, on average, the right connectivity, but obviously not precisely, right? You'll have some bundle of like scattered stuff, and then presumably the system would have to learn in maybe a few first hours or something to make sense of what they see, right? Somehow.
You're absolutely right. So there are multiple ways. It's hardly that you can make connectivity with a single neuron precision using this strategy. And it's actually designed to be sloppy, so you can accommodate, it can become robust. But there are multiple ways how that problem is solved. Yes. No question.
How does it resolve the information paradox? So this requires only one.
Exactly. So this is actually how this guy, Roger Sperry, why he formulated the information paradox. He said, look, there are only 2 molecules and I can make million connections. So he actually was a theoretician. Well, he was not a theoretician. He was a neural anatomist, but he a theoretician at heart. So he figured out that just with 2 molecules, you can create million connections. If you have gradients. So he predicted that there are gradients. He didn't know about them.
How does this create information? I don't understand.
It resolves the information paradox because you can make million connections. So there are million neurons connect to million neurons. And you can do trillion neurons, right, with the same strategy. But you only have 2 molecules, A and B. There are like 2 bits of information, limited number of information amount of information that you need to use.
You have not only two molecules, you have gradients who somebody directs, creates.
Right, but the gradient is also
This information. Where is this information?
this information is also easy to create. So, for example, what you can do is you can say, I'm going to look at the neuron at the bottom and multiply the level of this FB by 0.95 and express the protein on this level. And that will create exponential distribution like this. So it's information that doesn't scale with the number of neurons. It's a simple instruction.
That's absolutely.
Yeah, it's not like one bit of information, but it's a limited amount.
Because it's very sloppy. If it's sloppy, information is lost, right?
Right. So to answer the question how it's further made more precise, there are multiple tricks. Like you can have on top of those proteins, you can have another proteins which define it more precisely. So once you arrive to the zip code, you can do like more precise, like on the street level navigation, or then find a house. Or it can also be involving neural activity, right? You can refine connectivity using neural activity.
I mean, the newborn starts walking sloppily and then immediately learns. So there is some connections that are probably fixed afterwards in some sense.
Right. So
most of them are fixed after.
So there is like enormous number of
huge majority.
Right. There's an enormous number of tricks that the system is capable of using, and they all have to fit in within this 1 gigabyte. So we actually have been working on this model. I'm giving you like a very brief snapshot. We've been working on it for about a decade, and it goes back to like this is inspiration from the quantum Hall stripes. Turns out that some analog of quantum Hall stripes exists in the brain, and I'm showing here the picture from macaque monkey primary visual cortex, where you can see the black regions. So this is like a surface of primary visual cortex. The black regions are dominated by the left eye, and the white ones are dominated by the right eye. So there's this kind of zebra skin type pattern which resembles quantum Hall stripes, and we build the condensed matter like theory, which is basically based on the chemical labels that I described that describe this pattern. I'm not going to like basically describe you the details. I mean, if you're interested, you can read the details there. But it was a fun ride which lasted for about 10 years. But now enter the age of artificial intelligence, which I'm going to talk about next. Just to summarize, so the idea is that genome defines molecules, like it generates neurons. The neurons produce thousands of molecules. And also it defines the rules of how different molecules interact. They can be repelled, they can be attracted to each other, and maybe some more complex nonlinear interactions. And that ultimately leads to creation of the cortex, right? So we want to create an AI analog of this and like write a software package and apply it to different like sorts of AI algorithms. And this is how our algorithm works. So imagine that this is your cortical network. We call it phenotypic network. So it's already one that you want to encode in the genome. It's already created. And there is one entry in this like this matrix element ij in this network. So we are going to, for each neuron, for neuron I and for neuron j, we are going to define molecular labels, which are just from some array. It's probably like a 1,000-dimensional array, which defines all the proteins that this neuron can produce. And then we are going to send those proteins to the, we're going to send them to some small network, which we call GNet, which embodies the interactions between those different molecules. And this basically network will mimic the behavior of the genome. So this is generally our approach. And then we're going to use the various types of artificial intelligence tools so we're going to make this work perform at some task. So we'll require it, give it some X and require it to do some behavior, act in a certain way. And then we're going to use this backpropagation algorithm to learn the correct GNet. So it can perform using the input and output relationship. So this is the approach. So computer scientists are very sophisticated people. So they actually developed this idea. They implemented the chain rule of differentiation, and you can compute gradients. Like for example, you can compute the gradient of performance for this network, for the P network, which is your cortex. And then it will automatically compute the gradient for the GNet that will minimize this performance, that will optimize the performance. So you don't actually need to think about it. So it will happen automatically. But that's the overall idea. And remember those, like the red arrow is the neural development. So the neural development will produce the cortex. And instead of evolution, we're going to use this backpropagation algorithm that will shrink the cortical network into the GNet. So that's the overall approach. I'm going to illustrate it. In the example of this. This is a hydrogen atom for AI tasks, where this is like the handwritten digit recognition data set, where the network is you're supposed to train a network that can recognize digits written by American high school students. And so this is like the neural network. You feed it some image, like just like the pixels of the image, and it's a two-layer network. You know, there are some weight matrices, there are two weight matrices, it's a two-layer network. It actually be trained to recognize, it makes only 2% mistakes. So if you feed it 2, it will tell you 2. But, so what we're going to do is we're going to create this network from two networks, which are called GNets, which are 300 times smaller than the green one. And we're going to train those networks to generate this network, like from birth. And my point is that, so this is the example of performance for the. So this is a naive when you just train the network, the green network, to recognize handwritten digits from 0. It starts from 10% performance. Obviously, there are 10 digits. And then it slowly will train to 98%. But this is the one which is created from the GNet, from the blue network, from the genome that we trained. And at birth, it already can work with about 95% performance, despite the fact that it was shrunk by 300 times. So this is an example of bottleneck, right? So you shrunk the green network into the blue network by 300 times. And when you produce the green network from the blue one, it already can act at 95% performance. So it's as if you had some organism which was born, it can already recognize digits without any training. Yeah.
So to make the small GNet, you used an already trained full net?
No. No, it doesn't work actually. So if you just try if you fix the green net, the P net, it's not going to work. So you have to find a very specific P net that is compressible. Question?
How does it compare to just storing, like working out the neural network?
Yeah.
And then storing it with some just extremely lossy compression algorithm, and then starting anew from that?
Yeah, it's not going to work. I mean, with benchmark it, it's not going to work either.
So how do you make the small network? How do you?
This is like literally the structure of it. It's just a fully connected network. It receives for each neuron, it's a pair of neurons. It receives two arrays. In this case, it would be like X and Y. And it's literally, you can compute the number of elements in it, and it will be 300 times smaller than that one. I want to show you, so this is like a very important question, like what is actually going on. So this is like a space of all the networks, of all the PNets. So these are the networks that are high performance, and these are the compressible networks, the ones which you can compress into small genome. And what our algorithm is trying to find, it's trying to find this very small subset of the networks that are both high performance and compressible. So if you just take a random high performance network, you cannot compress it. It will be garbage. So I want to just to illustrate you, to entertain you a little bit, I want to look at the example of walking network. So this is like a physical simulator of something called half cheetah. This is like an organism which is limited, has 6 joints, and it's limited to move only in 2 dimensions. That's why it doesn't topple over, right? So this is a naive cheetah. It's controlled by like a small neural net, like 3 layers, about 300 neurons. So it hops around. It's a naive thing, not do anything. But the idea here is to train it to walk forward, right? So it's a typical. So this is a naive cheetah, and this is one which is trained to walk forward at the highest possible speed.
How you train it? I mean, what do you mean by training?
You train it to move forward at using some kind of algorithm which is called reinforcement learning. So you reward it for going forward at the highest speed.
Okay.
And this is the best you can learn. So it looks like it discovers this very weird solution, which is called reward hacking. So it chooses to walk on its chin for some reason. But like in 80% of cases, you will find that those solutions they have some kind of reward hacking. It will either like slide on its back or do something weird. And this is the cheetah this is the cheetah which is trained using our algorithm. It's about 5-fold compression. And in most cases, I think in 95% of cases, so this is a newborn cheetah, which is created from a smaller network, from a smaller genome. And it actually performs almost as an adult. You can see that it goes off the field a little bit later, but it moves almost at the same speed as an adult. So essentially, the organisms that can walk immediately after its birth. But the interesting thing, so what if we now put the two cheetahs into high gravity environment? So we put those two organisms in a novel environment. See if those solutions will generalize. So this is the cheetah which was trained without the genomic bottleneck. So it learned this reward hacked algorithm. So it's going nowhere. And our cheetah, it can still look on Jupiter in case of gravity enhanced by about 50%. It can still move. It looks a bit sloppy. I call it Jupiter shuffle, but it still works. So it just shows you that the compression of neural nets allows them to find solutions that can generalize across environments for some reason. We also show like more examples of this, which are very boring in our paper. I would like to talk about interesting thing, which I mentioned Newtons. I want to basically talk about how this may apply to physics. So this is like very conceptual and philosophical. So I learned from this book, Culture in the Course of Human Evolution, this idea that brains and culture, they co-evolve.
They do what?
They co-evolve, they form a niche. They evolve together. Human culture and brain, they evolve together, or in terms of that book, they form a niche, which means they kind of form like a negative imprints of each other. So one example would be the Chomsky hypothesis that the structure of natural language is somehow imprinted in the cortex. Another example is my favorite example is the acquisition of controlled use of fire. You know that when our brain consumes about 17 watts of energy, about 4 of those watts, they come because we cook food. The cooking gives us cooking took energy from the gut and gave it to the brain. So just by being able to control fire, we expanded our brain capacity by about 25%. So that's an enormous impact of culture on our cortical architecture, right? So arguably, Prometheus, when he gave us fire, he gave us 25% of our brain capacity. So this is important. So the question is if cultural phenomena can also form bottlenecks. I was talking about bottleneck in the brain, right? But I think like one interesting example is physics. So physics is condensing the behavior of the natural phenomenon, very, very compact rules, which can be described by equations and calculus, right? So, and the reason why this happens, like one of the reasons why this happens is because it's easy to teach, right? So you can teach people in like 12, 20 years, the entire content of our physics. So it's a way to pass this information from generation to generation. So this is an example, physics is an example of cultural bottleneck, right? So this is what this
science
department is. Any science which strives to find a reduced description of things, right? Simplified description of things. And in fact, the job of many theoretical physicists is actually to simplify things to the bare bone so that they can understand things, right? Anyway, so
What about misconceptions? I mean, also,
Excuse me?
if you have some misconception, you also can pass it through generations. So I'm not sure why it's about science rather than.
It's also a bottleneck, right? So it's another example of bottleneck, delusions.
Any theory.
Delusion.
No, they're not science, not theory.
Theories, yes. Yeah, as long as it's just
Bad theory, good theory, any. Religious.
yes. Okay, yeah.
I want to describe, I want to just notice that those two things, those two streams of information, like the brain, the cortex, and the culture can be viewed as self-replicating quantities. And the definition of self-replicating quantity is that if you wipe it out, it's not going to recover. Like if you, for example, forget the language, it's not going to recover, right? So the other stream of information genome cannot help you recover English language if it's lost. Or if, for example, some species lost, some cortical architecture, oh, wonderful, 5 minutes. If cortical architecture of some species is lost, there's no cultural phenomenon that can reconstruct, right? So each of those streams are actually independent self-replicating quantity or entity. There is another self-replicating entity which is emerging, which is tools. So tools are also self-replicating. For example, you cannot assemble a shuttle without a screwdriver, right? So if we lose all the tools, we will not be able to produce more sophisticated tools, right? So from that definition, the tools are becoming more and more complicated in the course of evolution, and they're co-evolving with everything. And I put here this modern GPU A100, which is also a form of tool, right? So, and tools should be added to this system of niches. So they also form a niche between culture and brain, right? So the point that I want to end this talk with is this, that in this triad of brain, culture, and tools, one can be easily gone missing, such as the brain. So those things can just continue self-replicating, to the future. So as a conclusion, I hope I argued to you that information about available in the genome is not sufficient to set up cortical connectivity with a single neuron precision. So evolution has to have discovered some set of methods of how to compress the brain architecture, some tricks. And we built AI tools that can help discover those tricks using just data sets, like large data sets that are available in AI. And and genomic bottleneck can help placing cortex through this genomic bottleneck and help force evolution to discover architectures which can generalize across environments and potentially lead to evolution of general intelligence. So I would like to thank my collaborators here and the funding, and you for your attention. Thank you.
Interesting. Questions?
Yeah.
Is there a reason why one cannot transfer the whole brain from generation to generation, and you have to first shrink it to a genome and then? Well,
this is how biology exists, right? So we go through single cell organisms to produce the offspring, and you can argue that it's a bug. I'm just saying that it's a feature. It actually helps us to become smarter. So that's the argument.
Yeah.
So you said that only some small fraction of possible architectures have the property that they can be compressed, right?
Yes.
And you have some ways to cycle through various architectures to find those.
Yeah.
Okay, once you find them, what have you discovered? And is there certain properties of those?
Oh yeah. It's fascinating. So like in that simple, and this is like a wonderful question. So our algorithm finds the architecture, but how does it compress? So it turns out that in this simple digit recognition algorithm, right? So if you don't involve bottleneck, the network will just remember the digits. So it will contain the weights in the network will just contain like masks corresponding to the digits. So if you start compressing it, it discovers the Fourier transform. So the first layer of the network will do some analog, will do Gabor transform, like a local Fourier transform, because the digits are kind of translationary. You have to recognize them in different points. And then the next layer will learn how to deal with the Fourier transformed image. So this is fascinating. You have to actually do neuroscience on those networks that we'll discover, and we're only scratching the surface of it.
So you're talking about the connections and the spatial architecture, but the information also exists in time. It's the timing. You'll have neurons that are all firing, and then suddenly they'll form a coherent oscillation.
Right.
Will then disappear. And so that part also, I don't know, would that part be part of the culture? Because that's something that you could have you could know all of the connections, like knowing a map of every road in the United States still won't tell you how long it'll take to get from one point to the other because there's traffic and other issues.
Yeah, absolutely. We're only scratching the surface of it. And the simple organism, the half cheetah, it does actually process information in time, but we don't know how it does it. So it is somewhere hidden in the network, and we need to do neuroscience. But I agree with you. Like the way how brain deals with time is very complicated. It has a recurrent neural network, which has intrinsic time scale, multiple intrinsic time scales, and that's how it can do it. And so it's too early for us to really see how it's going to work out if we try to bottleneck some arbitrary recurrent neural network.
Okay, so
Thank you.